All right. So a couple of things. I don't know if folks saw the announcement or not, but there's a stem cell training. It is free if you want to go. It is March 3rd over at Madison College. It's basically how to work with stem cells. Dr. Kretschmar is going. I will not be going because I have other plans that day already. Holy plans. So let me know if you want to go, and I will email him and let him know. They need a certain number of people to run the course, so sounds like it's an all-day fun thing. But anyway, all right, I'll leave that at that. I think that's it for right now. What I wanted to start with is actually take a little bit of time today to go through the critical thinking problem and walk through it together. But before we do that, I think I might need to review this real quick, restriction enzymes. This is all the way at the end of the slides, but I figured this is good time as any to review it. So I guess to give you a historical perspective, restriction enzymes, we use them nowadays as a way to analyze DNA. Anybody know what their function in nature is, though? Or where we even get, where they come from, Sabrina?
They're from bacteria, and they eat up all the foreign bacteria and stuff.
It's an in some respects, it's an antiviral response. These are used to chop up or break down phage DNA. And we'll talk about how phage, one of the ways in which phage can overcome that, and once we get to that section. But in the context of what we typically use these for is as a way to analyze particular sequences within a fragment of DNA. Before sequencing came about, this was kind of a gold standard, and actually in some respects it still is a gold standard for analyzing bacteria that are involved in outbreaks. So you would take their genomic material, chop it into fragments, recognized by a restriction enzyme. For this example here, GAA-TTC is the restriction enzyme site for EcoR1. The first three letters tell you the organism, E. coli, and then R1 refers to some additional nomenclature. But this one is one that cuts and leaves these sticky ends. If we were to look at it, that's more info than you need right now. But once we analyze it on a gel, or once we run it onto a gel, we should see different size fragments based on whether or not these restriction enzyme sites are present within the DNA. And so that takes us to the critical thinking problem. Who struggled with the critical thinking problem? You can raise your hands. It's okay. Good.
We did.
Good. You should have. This is about the time where we all need to start pushing each other. Okay? And I don't say that as a bad thing, but the only way we're actually going to learn anything is if we start pushing each other. So that what that problem was designed to do in part is to start getting you to think about, holy crap, how am I going to make it through 11 more weeks of this? Just try, okay? Try. We'll figure it out together, but sometimes you've got to be challenged a little bit. All right, so in this particular problem, I walked through this with a couple of people yesterday, and I think one of the big things is that I feel like you guys actually know more than you're letting on. And this is where I'm trying to get at the, let's start pushing each other a little bit. You know the information, but I think it's the how do you actually apply it, or how do you recall some of that information to help inform decisions where we're maybe struggling right now. And that's what we need to start working more on in class. So let's just walk through the prompt on this problem, and let's just start by listing what do we know about this virus. It may not be related to the problem, but let's walk through it and see, pick out some key terms. So, sin nombre virus. Who takes Spanish? Grace, what does sin nombre mean?
Nombre is name.
What does syn mean? Without name virus, okay? A member of the hantavirus genus is an emerging pathogen in the U.S., carried by rodents in most cases in the U.S. and has occurred in the Four Corners region. The viral genome is segmented, negative single-stranded RNA, encoding a viral polymerase, a nucleocapsid, and a heterodimer glycoprotein. Why would this one have to encode a polymerase?
RNA.
It's an RNA virus. What does it mean that the genome is segmented?
It's not just one piece of RNA, it's a couple.
Good. It's at least more than one fragment of genomic material. And then negative single-stranded tells us a little bit about it as well. What has to happen before translation can occur? Good. So this one here would be an example of one where we go from negative single-stranded RNA to plus single-stranded RNA, and that then can be translated into protein. And also we know, it might not be the same, but it also has to produce full-length single-stranded RNA for the process of replication too. Those are things that we can figure out right away. Annie?
Quick question.
Yeah.
If it's segmented, then does it just create a single-stranded RNA for each one and then?
It would have to, yes.
Okay.
Yes, absolutely. All right, you've been able to map 2 mutations with the glycoprotein, the GCGN, that render it incapable of producing infectious variants. In other words, there's some problem along the way that prevents the life cycle from occurring. This is a glycoprotein, meaning that it's on the surface of the viral structure. What are potential roles for it? What could it be involved in?
Receptor protein.
It could be the receptor protein or the spike or the receptor binding protein. What else? What other steps might we see glycoproteins being involved in here? Entry, or the fusion events maybe. So maybe entry, maybe uncoding. All right, those would be the major ones at least. And then you choose a recombinant VSV system, which we haven't covered this, and I realized that after the fact. So what is this? VSV, this is the structure. Capsid, and it's enveloped. It looks like a bullet. The recombinant VSV system is one in which you can take the VSV genome, and this stands for vesicular stomatitis virus. You can take its genome and you can put into its genome genes of your choice. In this particular example, what the researchers are doing are putting in the genetic sequence encoding the wild-type GCGN, or encoding one of the 2 mutants, which we're calling G1 and G2 here. So they generate 3 different types of viruses. This system is actually commonly used to study attachment and entry in vitro. And what I mean by that is Ebola. Ebola, as soon as we say the name, it strikes fear in people. It is a BSL-3 organism, requires basically a spacesuit to work with it. You can study the attachment and entry in isolation by taking its glycoproteins and putting it into this VSV system. That changes the safety level. That changes how much training it takes for individuals to have to get before you can begin to work on it. And then you also have here a GFP protein to measure expression of the genome. So this would be one where green fluorescence protein is put into the genome as well. As soon as you get RNAs being transcribed and then being translated, you're getting GFP as a way to detect which cells have truly been infected. So this would kind of be like, if you were trying to track which cells are infected, it'd be kind of like an immunofluorescence. All right, similar idea. All right, so with that set up, What kind of blot?
Western.
Western. Something we haven't talked about so much yet is oftentimes with Western blots we have a standard that we use to determine the size of the proteins. Wild type, G1, and G2. There's two parts to this problem. One, describe the results. Two, interpret the results. How would we describe what we're observing? What's happening with the wild type? Two separate proteins, sizes? 75 and 50 kilodaltons, roughly. What about G1?
130.
And just a single band, right? And then G2? Same as the wild type. Okay? So which one of these has a mutation that we can detect by Western blot? G1 has the mutation, okay? What is the mutation? We can't necessarily say what sequence the mutation is, but maybe what the effect of the mutation is on GCGN. GCGN is a heterodimer. Two proteins that bond together to form a protein with a slightly different function. What's not happening in this G1 mutant?
Cleavage of the two proteins.
Cleavage of the two proteins. This is a polyprotein. Okay. Now, is that the right answer? Yes. Is it the only answer? No. What I mean by that is I'm not worried so much about you guys being able to say it is definitely this because this is what we talked about in class. What I'd rather you be able to do is say, I think it's this because the data supports it. Okay, use the data and use the ideas that we've talked about to help make these decisions. So for example, Shania, what supports the idea that it's inhibiting cleavage?
Because like the KD of G1 is about the summation of the two.
Yeah. So that 75 and about 50 leads to a product that's 125, 130 kilodaltons. That would definitely support it. Here's some additional evidence. We're using a polyclonal antibody, and what that means is this is an antibody. It's kind of like a cocktail of antibodies. So this is my really crappy rendition of this polyprotein. A polyclonal antibody means we've got different ones that are binding all over the place on this thing. Okay? And because this polyclonal cocktail can detect still the protein in G1 and G2, what this is telling us is these must be the same protein, but that there's a defect or some sort of mistake, cleavage being one example, that is rendering this inactive. What are other experiments that we could use to say that, yep, these two are produced from a polyprotein that undergoes cleavage? What else could we do? Annie?
Protease inhibitor
So, what is a protease inhibitor?
inhibits the host protease from cleaving at that point.
Yeah, absolutely. So if we used a protease inhibitor here to block the cleavage, what we would expect to see is a single protein band about the same size as G1 there. Absolutely. And that would additionally support the prediction or hypothesis that this defect here is a result of inability to cleave the protease, or to be cleaved by protease. What else could we do? What other things could we do outside of even Western blotting?
Immunoprecipitation?
How would immunoprecipitation work here?
Even if they did get cleaved, would they stick together?
That's a really good question. In this case, they probably would, since they're a heterodimer, meaning that they typically will bind even through non-covalent interactions with each other. I see where you're going with it. It's a good idea. It may not work for this one. What's going on?
Cream right there. Just like,
So in this particular problem, we're cutting with small 1. How many bands do we get for the wild type?
3.
Okay. Sizes?
2,200, 1,000, and 400.
Works for me. Anybody as kids play those or have those books where it's like 2 pictures and there are 10 things wrong with one of these pictures. That's exactly what we're doing here. That's exactly what we're doing. So, here is your original picture. What is different from the original picture? Please humor me.
G2.
Thank you. G2 gives us 2 bands. What size? What size is this? 2200 and 1400. 1400 is roughly the size of what? The 2 smaller bands in the wild type. So if we were looking at this, what we would likely predict then is that there's a mutation that's inhibiting the ability of this band to be cleaved into 2 smaller ones by a small one, right? If we'd go and sequence it, yes, there's U's here, that's my mistake. But I'm guessing the mutation most people recognized is, hey, this C gets turned into an A. And if you take a look at this, what does this do to, let's start with this. What does this do to the small one site? What is the small one sequence that it recognizes? CCC, GGG. So it goes from that to ACC, GGG. Obviously that's not going to work. What effect does this have at the amino acid sequence? Proline to threonine, and properties of those two? Non-polar to polar. And at least in this example, then, what that says is it's inhibiting the process of fusion. Okay. So that's how we would walk through this. I feel as if you guys know how to analyze this. I don't know if it's hesitation or just like Linus in a security blanket sort of thing, but I know you can do this, okay? You get another opportunity to on Friday when we read a paper. Sabrina.
So would the non-polar to polar affect pH dependent?
Probably, yeah. Yes, is the answer in this one. All right. Grace, go ahead.
How do you know if an amino acid is polar or non-polar?
You look at a chart that tells you.
Okay.
Simplest answer. I would not expect anybody to be able to know that off the top of their head necessarily, unless you've had Kretschmar who has forced you to memorize all the amino acids. I know like some, but not all. And I know if it's got an OH, it's typically polar. Proline's it's kind of connected to the backbone. It creates really sharp turns in the amino acid sequence, and that structure of it is all carbons and hydrogens, so it has no polarity whatsoever. So really it's the side chain that you're looking at when we decide is it polar or non-polar. All right. I was thinking about going in for us doing another problem, but I think I want to skip around a little bit so that I give you guys at least enough information for Friday's paper. So I think that would be a better use of time today. So what we're going to do is we're going to talk for a few minutes about plaque assays and other ways that we can determine is a cell actually infected with a virus, non-plaque methods. And then we'll try to get a little bit into PCR and real-time PCR and reverse transcription as kind of a precursor. Plaque assays are a commonly used method to analyze concentration of viruses. They work only if the virus will actually lyse the cells that it infects. And there are modifications to this as well. There are other assays called foci assays where you're looking for actually an overgrowth of cells. But we'll maybe come back to that at a later step. Many in here have taken micro, not everyone, but one of the things you may remember is that one of the common ways we determine concentration of bacterial cells is from an original culture, perform a serial dilution, and then take those dilutions, plate them, and then we can perform some calculations to figure out what the original concentration of the bacteria is in that culture. We do the same thing for a plaque assay, but there's a slight modification here. Again, viruses require a host cell, so we need to provide that for them in order to visualize the concentration. So what we do in this experiment is we take our, in this example we're just going to refer to phage here, we take our bacteriophage, dilute them down in 1 to 10 dilutions, and then after that we take 100 microliters of each of them, mix them with 100 microliters of bacterial cells. The volumes are not important. I guess I'm just saying that because that's how my lab does it. But equal concentration of, or equal volume of virus and bacterial cells. Stick them into the incubator for 20 minutes. That gives enough time for attachment and penetration. May even allow enough time for the viral life cycle to begin. So replication, biosynthesis. And then afterwards, you take your sample out and you pour out onto the plate the bacteria and the phage together, and hopefully what you observe the next day are plaques. From one of these plates, you're going to count the number of plaques, and to figure out the concentration, we need to know PFU, or plaque-forming units. We need to know the volume of bacteriophage that we plated. For me, it's typically 100 microliters or 0.1 mils. And then the final thing we need to know is the dilution. From which dilution of the phage did we count the number of plaques? And using these 3 units then, we can calculate the concentration. So a simple calculation is just PFU divided by the volume times the dilution.
You said the volume of the phage?
Yes, yes. Now the reason why we're going over this is because actually on Monday, remember, you got to bring a sample with you that you think might have Pseudomonas fluorescens phage in it. I'm going to have you try to calculate this on Wednesday from your sample. It may be that there's nothing. That's okay. We got backup plans. Bill's going to totally sabotage the experiment, just so you know. All right. So here's what it looks like in a case when you've got these plaques. I won't have us do this problem right now, but if you wanted to, you would be more than welcome to analyze this. You can see in this problem from Rachel Butts at Hope College. This is a mycobacterium phage that they named Aurora, and you can see e to the -6. That means the dilution is 10 to the -6. We'd assume that they're plating 0.1 mils, and if you counted up all of these plaques on here, you would be able to get the concentration of this particular phage in the original stock culture that they're working with. Okay. Roughly, if you do the math correctly, it's going to come out to somewhere around 10 to the 9th per milliliter, roughly. In eukaryotic cells, we have to do these plaque assays a little bit differently. And the way that we do this with eukaryotic cells is that we actually, since eukaryotic cells are not easy to see, like a lawn of bacteria, we have to stain them. So you would perform an experiment similarly, in that in this case what you're doing is you're adding adding a virus to a plate of human cells that are flat on the plate. You also add maybe a thin layer of agar or another type of solidifying agent that would prevent the phage from, I keep saying phage, prevent the virus from freely floating on the plate. And what that allows for then is for the virus to infect a single cell, replicate, infect those around it, replicate, so on and so forth, producing these plaques. You then stain this with a dye like methylene blue, which helps you visualize where those plaques are. And those plaques are those empty spaces where there are no cells. This is what we call, in this particular one, this would be an experiment in which we are testing 2 different viruses. on a monolayer of eukaryotic cells, and we're testing their ability to infect in the presence of a drug. Okay? So you can assume that the drug concentration is increasing from left to right here in each of these, and you'd be able to determine, for example, what concentration of the drug inhibits 50% of infections. One of the ways that we can analyze this right now is just by saying, which of these two viruses is inhibited by the drug?
One.
Okay, good. You know how to read this. And if you wanted to determine what concentration of the drug inhibits 50% of the growth, you just got to count the number of plaques and then do a little bit of division. Jonah.
What kind of media are these eukaryotic cells grown in?
Ah, that's a great question. The media, so eukaryotic cells, we have two types that we often work with. Adherent and then ones that are more planktonic or free-floating. Your free-floating ones are going to be your white blood cells, B cells, T cells. They will remain suspended. Other ones are going to adhere to a surface. And so oftentimes to grow human cells in culture, you have to use specialized petri dishes that have been tissue culture treated, which allows for those cells to grow and basically adhere to the plastic on the plate. The media you add to them, the most common one is known as DMEM, which stands for Delbecco's Modified Eagles Medium. And it's got nutrients, it's actually got phenol red in it as a pH indicator, because you also need to provide a certain concentration of CO2 to keep these at homeostasis. You're also adding something like bovine calf serum, or that's the cheaper one. Bovine fetal bovine serum is more expensive, but those will provide additional nutrients that's found in blood serum that would allow for these cells to grow. So basically growth factors. Typically, these cells will grow to confluency, meaning they cover the full plate, and then at that point you would infect them with the virus. Yeah.
Infecting them, or do you just spread virus?
Yeah, so what you would end up doing, or I guess when I was in graduate school, if we wanted to do an infection with EBV, what we would end up doing is taking a 6-well plate like this one, and we would wash out all of the media that was on that confluent layer of cells. We'd add back some fresh media without fetal bovine serum because that seemed to inhibit infection. So you add in everything but the FBS, you add in your virus, and then you incubate it at 4 degrees for about an hour or two. And that cold temperature actually improves the attachment step. And then 24 or 48 hours later, after you have incubated these cells and the virus at body temperature with 5% CO2, at that point then you would stain them and look for plaques. Sabrina.
So if the viral egress was budding or exocytosis, this
You're absolutely right. This wouldn't work if it were a budding mechanism. And so that leads to different types of techniques. This obviously is the easiest of them to do. The other ones, what we're looking for are what we call cytopathic effects, or CPEs. So that's a great leeway into this next part. As Sabrina just pointed out, some viruses we can't detect by a plaque assay, and so we have to do more microscopic techniques to identify what we call these cytopathic effects. Typically, what's measured here is what's known as a TCID 50. I really don't want you to know anything more than it's kind of like a modified plaque assay. There are techniques to determine the concentration or infectious dose of a virus using these cytopathic effects as a measurement. And the TC stands for tissue culture infectious dose. We'll actually see a modification of this for the paper we're reading on Friday. They use a measurement known as CCID, which is cell culture infectious dose. It's the exact same thing. But it's a measurement of the concentration of virus within a sample. So what are CPEs? Anything abnormal. Anything that doesn't look right. Inclusion bodies or viral factories that we might be able to even see with a light microscope. Rounding up our loss of adherence of cells, as is being shown here. So these are, I can't remember which cells these are, but they're being infected with a virus known as SV40. And you can see here's what the cells normally look like, and they round up, and it changes the refraction of light as they end up rounding up. And so those represent actually a couple of the cytopathic effects that we can see. Rounding of cells, changing in the ability to refract light with a light microscope. We might see granules or other abnormal looking staining which indicates viral factories. This one also exhibits loss of adherence. And then other ones like this right here. What do you observe in this image? Oh, by the way, this is a single cell. What's all that red stuff for those oblong structures in the middle? I'll point out in contrast, here is another cell. Here is another one. What was that?
The nucleus?
It is the nucleus. All of a sudden, this has become a multinucleated cell. How the hell did that happen? Well, basically what we get is cell-to-cell fusion. The viral proteins that are required for fusion of the viral membrane to the host membrane, as that virus is undergoing biosynthesis, those proteins are being exported to the cell surface. And if we get two of those, so here's the viral protein on a cell, here's its nucleus. Obviously, these still have to bind to the cell, to the host cell receptor. Bless you. We can actually get fusion between these cells, creating multinucleated cells, or what we call syncytia. This word right here. So that's another sign of, or another cytopathic effect. Grace?
So these, all three of those show the concentration of a virus?
So you can use these as a way to estimate the concentration of a virus, or what they call the TCID50. And it's really based on a measurement of how much do you have to dilute the sample to see these cytopathic effects in 50% of your cells. That's roughly what it means. Now, not all of these are going to take place in every single virally infected cell. Some are specific to certain types of viruses based on their life cycle. All right, that takes us through detection. What we're going to focus on when we do our experiments will be plaque assays. But do know that this TCID50, if you come across it, or CCID50 in the paper, again, it's just a measurement of viral concentration. Okay. Last thing to talk about, or last thing we'll cover today is a bit about nucleic acid detection. We've already started talking about some methods, and one of the methods we're going to actually use when we work with bacteriophages, we're going to do some sequencing. That's kind of the newest of these approaches. And we'll talk a little bit about that. We're going to talk about 3 PCR-based ones, and we're going to kind of go from, like, in order not only of one word that they created, but also from the simplest to the most complex in terms of what can they tell us. And then if there's time, maybe we'll spend a little bit talking about what a microarray is as well, and how we can use that to measure mRNA expression. What is PCR?
Polymerase chain reaction.
That's a great start. Nice knowledge-based question. You would win $100 on Jeopardy. What does it mean? Describe the process of PCR.
process?
Sure.
Both use like a thermocycler versus like denatures the DNA, so it just like unwinds it, and that allows for proteins to build new strands of DNA. Just get a ton of
What proteins, Jonah?
DNA. Polymerase.
What kind of polymerase?
DNA polymerase.
Okay, Taq polymerase. Good. What else do we need in this reaction? What else do we need in a PCR reaction?
Nucleotides.
We need a substrate, nucleotides. We need primers because every DNA polymerase requires two things: a template and sequence to begin from. Those are kind of the major ones. The rest are things like you need your template DNA, you need magnesium as a cofactor. What else are we? Nope, those are the big ones. Polymerase, oh, and buffer. Basically, replication in a test tube, right? And if you think about the process of replication, okay, I'm going to force you to go back to Gen Bio and other courses for a minute here. Genetics, I'm sorry. How does replication occur? We have separation of the two DNA strands. What helps for that? What enzyme is required for that separation? Gyrase is involved in uncoiling it. The other one that we might say is involved is helicase. I think I heard somebody say as well. Why don't we have helicase in a PCR?
It's a denature from the high temperature.
We've got temperature to deal with it, exactly. And then we've got polymerase moving in two directions from a RNA primer that's generated by primase and replication. And in the case of DNA, or in the case of PCR, we just use DNA primers because they're a heck of a lot more stable. And it also allows us then to detect the specific part of DNA that we want. Here's a typical cycle. 95 degrees Celsius for 10, 15, 20 seconds. That denatures the DNA, as Jonah pointed out. Drop the temp down to about 50. That allows for the primers to anneal. And then 72 degrees for however long you need to amplify the DNA. It used to be a rule of thumb that every 1 kb of DNA you were replicating, you needed to let this amplification step go for about a minute. Nowadays, it's like 10 seconds is what they suggest with some of these enzymes. And again, you need a thermostable polymerase. Taq polymerase is great because Thermus aquaticus, the bacteria that gives us that enzyme, is a thermophile. It grows at 72 to 80 degrees Celsius, so it can handle that temperature fluctuation really well. Okay. What is the goal of PCR?
Amplified DNA.
And not just any DNA, but a specific fragment of DNA, one that is of your interest, right? Is it quantifiable? Yes, if you include real time, but let's take that out right now. Just PCR itself, is it quantifiable? Annie?
Isn't that like semi?
It can be, but you have to have you have to be able to stop after so many cycles numerous times to be able to do that. You're absolutely right. Oftentimes, though, how is it used? It's often used as a dichotomous answer or a binomial answer. Yes, your DNA is present. Nope, it's not there. Yes, it's this size. Nope, it's not that size. Okay. Theoretically, each round of replication should give us how many new fragments of DNA, or new copies?
Double.
It should be double. From every 1, we should get 2, and then from 2, we should get 4, and so on and so forth. After 30 cycles, we're talking on the order of billions of copies. But that's not what happens. Okay? Like communism, sounds great in the books, but in reality, not as great. Speaking of communism, did anybody watch ice dancing last night, or ice skating last night at the Olympics? Sorry, it wasn't dancing. It was the pairs figure skating. The Democratic Republic of Korea, North Korea, had an amazing show. But anyway, I digress.
That's actually the same.
Yes, it's a real thing, guys
Um,
and gals.
and gals. You do it in your free time.
Yeah, in between watching Shaun White and other things. So. All right, PCR. Theoretically, should be exponential. 2 to 4, 4 to 8, so on and so forth. That's not what we see. Oftentimes we see this. It peaks. Why? Why does it peak off? That's an option. That's a possibility. Nucleotides are limiting. That's not the only reason, though.
Do proteins denature from all that?
That's another option. That's not the only explanation, but that definitely can contribute to it. Grace?
Maybe not enough nutrients and support too.
It has something to do with that. It's not nutrients, but it's actually you get a buildup of an inhibitor. So one dirty little secret of this process, we have deoxynucleotide triphosphates. In the reaction of adding nucleotides to genomic material, this gets broken down into what will be released. It's what's known as pyrophosphate, or 2 phosphates bound together. This right here is an inhibitor of DNA polymerase. So in part, what you're observing here, this loss of linearity is, as Jonah pointed out, DNA denaturation, as Amelia pointed out, nucleotides potentially running out. And the third is you've got an inhibition of the process. Now typically when we do PCR, just traditional PCR, we're looking at maybe 35 cycles out, we're looking at an endpoint analysis. And this is well beyond the linear slope. And so that's where techniques like real-time PCR have become really powerful. Instead of having to, Annie was pointing out, you could make this quantifiable or semi-quantifiable. You could set up a PCR reaction, or 8 of them, identical, and say, I'm going to take one out after 5 cycles. I'm going to take one out after 10. I'm going to take one out after 15, so on and so forth. So that you can make sure that you identify your DNA concentration within this linear range here before you slope off. This is time consuming. It's labor intensive. So let's just throw some money at the problem. Real-time PCR. And it's such a powerful technique. I think what we'll do is we'll stop there for today, but I think what we will do at the beginning of discussing the paper on Friday is just cover real-time PCR briefly, because they actually do use it. They use it to quantify the amount of viral genomes at some point in the experiment. So we'll take time to do that, and then we'll come back to RT-PCR probably on Monday, all right? So paper, submit questions by Friday class time so that we can discuss BYOM. Anything else I'm missing? All right, have a great rest of the day, everyone.